Здравствуйте, глубоко уважаемые.
Good afternoon, dear colleagues and guests. Let me declare open the session of the Dissertation Council for Defense of Thesis by Lopachev, Alexander Vasilievich for the degree of Candidate of Biology, specialty 145 Biochemistry, study of mechanism of neurotoxic action, Cardiotonic, Steroids, ouabain, digoxin and bufalin on the primary culture red neurons. Today is the 27th of December 2021, 3:05 Moscow time. The order of Academic Secretary of St. Petersburg University, note of Alexander Valentinovich, dated with 27th of October 2021, number 102885/1, me, Shamov Olga Valerina, Doctor of Biology, Associate Professor, Head Professor of the Department of Biochemistry of St. Petersburg University, was appointed Chairman of the Dissertation Council. Other members of the Council were appointed in the same order. Let me introduce them to you. Klimenko Viktor Matveevich, Doctor of Medicine, Professor, Leading Researcher, Laboratory of Neurobiology of Integrative Brain Functions, Honorary Doctor of Institute of Experimental Medicine. And in the remote access mode, we have Antonov Sergei Mikhailovich, Doctor of Biology, Head of the Laboratory of Comparative Neurophysiology, Section of Institute of Evolutionary Physiology and Biochemistry, Russian Academy of Sciences. Can you hear us? Sergei Mikhailovich can hear us. Salmina Alla Borisovna, Doctor of Medicine, Professor, Head of the Laboratory of Experimental Neurocytology of Brain Research Unit, Research Center for Neuroscience. Alla Borisovna, can you see? Can you hear us? Yes, I can. Thank you. Nikitina Natalia Nikolaevna, Doctor of Medicine, Honorary Senior
Отделение биомедицины.
Research Fellow, Department of Biomedicine, Cardiff University, United Kingdom. Ninkina Natalia Nikolaevna. Yes, I can hear you. Thank you. And the degree applicant, Lopachev Alexander Vasilievich, is also present. For reason, the academic supervisor, Fyodor Tatiana Nikolaevna, Doctor of Biology, Head of Clinical Experimental Neurochemistry Laboratory, is absent. In order to improve the quality of communication, dear colleagues working in the remote access mode, please switch off your microphones, but remember to turn them on when you're given the floor. Thank you. Let me also inform you that our session is being recorded and broadcast live at St. Petersburg University website. The speeches are being simultaneously translated from Russian into English or from English into Russian. During the live broadcast, an email is currently displayed to which all the listeners may send questions to the applicant and express their opinions regarding the content of the thesis and the current scientific discussion, thus taking part in it. These questions shall be forwarded to me by our technical support department, and I shall present them during the discussion. Questions should be related strictly to the applicant's speech and the content of his thesis, and it's necessary to indicate full name, position and place of employment of the author. Questions that are not related to the scientific discussion of the text and assessment of the thesis, as well as anonymous questions, shall not be presented. In accordance with the order of awarding academic degrees at St. Petersburg University, approved by the local regulations of St. Petersburg University, hereinafter the order, a session of the Dissertation Council shall be considered competent if at least 2/3 of the appointed members are present, but not less than 4 persons. Our council consists of 5 members. All 5 are present, including 2 council members, 3 council members working in the remote access mode. Order and visual contact has been established with them. Thus, we have the quorum. I invite the Dissertation Council Activities Support Department Officer to draw up the attendance list. Let me set forth the following procedure of today's session of the Council session, the approximate duration of 2 hours. Summary report of the Chairman about the applicant and documents submitted by the applicant and their compliance. Answers to possible questions, approximately 5 minutes. Applicant summary report outlining the key points of his research, approximately 20 minutes. Questions to the applicant, strictly on his report, no more than 2 minutes per each question. Answers of the applicant, no more than 5 minutes for all questions. Speeches of all the council members in turn with their assessment of the thesis and the applicant's speech, as well as questions and suggestions to the applicant, approximately 10 minutes per speaker. Speech of the chairman with his assessment of the thesis, approximately 10 minutes. Answers of the applicant to questions and remarks made by the council members, no more than 20 minutes. Open discussion. Speeches of the attendees with summaries of their opinions and of specific questions and suggestions to the applicant, strictly on the topic of the study, no more than 2 minutes. At that, all the speakers are requested to register themselves and fully introduce themselves before speaking, given their full name, position, and place of employment. Next item, our program, presentation of questions to the applicant received during the discussion and live broadcast of this session via St. Petersburg University website. Let me inform you that written questions, the presentation of which they shall take more than 2 minutes, shall not be presented. Answers of the applicant, no more than 2 minutes per question. Speech of the academic supervisor, not more than 3 minutes. A discussion before voting. For that period, sounds shall be switched off for approximately 5 minutes. Open individual voting. Votes shall be counted by the chairman. The results shall be recorded in the minutes of the meeting. Make a decision on awarding or not awarding the degree. Closing remarks of the applicant, no more than 2 minutes. Dear colleagues, do you have questions or objections to this procedure? If there are no questions, let's proceed. And first of all, please turn the sound of your mobile phones off. Dear council members working in the remote access mode, please keep your phones switched on so that we could reach you in case of emergency. Thank you for your understanding. Let's start our session. Lopachev Alexander Vasilievich wrote his thesis for the degree of candidate of biology, specialty 154 biochemistry, on the theme study of mechanism of neurotoxic action of cardiotonic steroids, ouabain, digoxin, and bufalin on the primary culture of rat neurons. Was accepted for defense by the order of academic secretary of St. Petersburg University, dated the 7th of October 2021, number 9604/1. Lopachev Alexander Vasilievich wrote his thesis at the Neurology Research Center under the supervision of Doctor of Biology, Head of Laboratory of Clinical Experimental Neurochemistry of Scientific Neurology Scientific Center, Fedorova Tatiana Nikolaevna. The number of publications which set forth the key findings of the thesis, according to the enclosed list, is 5 in the journals from the list approved by the Ministry of Science and Education, Russian Federation, in journals indexed in scientific databases, Web of Science and Scopus, 4 publications. All the documents submitted by the applicant, according to the information, are received from the curator, comply with the requirements of St. Petersburg University, and are kept in the applicant attestation file. Copies are also available from the Dissertation Council Activities Support Department officer, who is currently present. Before I give the floor to the applicant, dear council members, do you have any general questions to the applicant, and is it necessary to review the entire list of documents submitted by the applicant? Do you have any questions, and is it necessary? No. Then, no, we don't have any external reviews. Let me remind you that the applicant may answer each comment on each review after the review is presented. So do we need to discuss that now, or should we first? So first the applicant will speak. Okay, Alexander Vasilievich, the floor is yours. Please present the key points of your research. Distinguished members of the council, allow me to present the thesis for the degree of candidate of biology in biochemistry, study of the mechanism of neurotoxic action of cardiotonic, steroids, ouabain, digoxin, and bufalin on primary cultural rat neurons, carried out under the supervision of Doctor of Biological Sciences Tatiana Nikolaevna Fyodorovna. Cardiotonic steroids are used as medicine for cardiac failure, and their raw material is mostly made of plants and toads. Cardiotonic steroids are specific inhibitors of potassium, sodium AT phase, are present in the central nervous system and consist of 3 subunits: alpha, beta, and gamma, and they transport potassium ions out of the cell and sodium ions inside the cell. In the central nervous system, there are 3 isoforms of subunits. Alpha 2 is present in all types of cells and supports sodium, potassium as and is needed to remove excessive sodium from. And alpha 3 is present only in neurons, and its main role is restore sodium, potassium. Right. The sodium, potassium AT phase corresponds with exchangers, glutamates, as well as receptors, ionotropic receptor, dopamine receptors, and many others. In addition to cardiotonic steroids, there are many factors that affect sodium potassium AT phase, which can be divided into oxygenation stress and also the signal functions. These are various kinase, as well as cardiotonic steroids, which, unlike those listed above, are specific inhibitors of sodium potassium AT phase. What is important, besides changing the sodium potassium activity, its inhibition may lead to degenerative processes. Thus, we may say the following prerequisites. So there are endogenic steroids, which an increase in their number was detected in case of various pathologies. There are side effects for the central nervous system. There's a heart failure, such as digoxin, and toxic for neurons, ouabain activates a number of signal cascades in different cells, and ouabain neurons activates. Thus, there is a question: if there are differences in toxicity of different cardiotonic steroids, and what are the mechanisms for toxicity? The purpose of the work is to investigate the neurotoxic effect of cardiotonic steroids, ouabain, digoxin, and bufalin on primary red neuron culture. And to achieve this purpose, we set ourselves the following task: to compare neurotoxicity of ouabain, bufalin, and digoxin as in primary cultures of red neurons, to compare the effects of CTS, of ouabain, etc., on reparations of adult red brain major hemispheres, to study the effect of ouabain, etc., inhibiting alpha-3 and alpha-1 isoforms on activation and temporal profiles of activation in primary cultures of red neurons, determine whether the activation of each kinase is dependent on changes in the cellular concentration activation of other kinesis when ouabain acts on neurons, to identify the relationship between the increase of the cellular concentration and the toxicity, identify the relationship between changes of kinase activation and ouabain toxicity in primary culture red neurons. In order to solve these tasks, we use the following methods. To assess neurotoxicity, we conducted a test and with in fluorescent microscope, and also in order to assess activation of signal cascades intercellular, we analyzed with the ratio of the phosphorylated form of kinase to the total kinase form. This part of the work determined the red large hemisphere brain, and to measure the activity of sodium potassium ATPase, we in preparation obtained of red large hemisphere brain, we measured the speed of formation of non-organic phosphate during disintegration of ATF. We compared neurotoxicity and in ouabain, is a concentration which became the minimal 24 inhibition of the culture, concentration of digoxin is under the same conditions, and for bufalin is toxic at more than 1 mole per liter. Thus, we assessed the impact of toxic and non-toxic concentrations on the rate of anti-apoptotic to pro-apoptotic family proteins. And we indicated that the ratio changes towards dominance, which may mitochondrial apoptosis pathway. And we measured the activity of sodium potassium ATPase, which included alpha 1, alpha 2, alpha 3 isoform activity, and we created curves of bufalin and digoxin with the help of mathematical decomposition of the curve into biofunctions. We found that sensitive to ouabain alpha 2 and alpha 3 by activity have about 15% of total and 85% of alpha 2. So we determined constants for ouabain, digoxin, and bufalin. And as you can see, for ouabain, the coefficient as a constant for alpha 2, alpha 3 are very different by 3 orders. For digoxin, we see more or less the same picture, but the bufalin has a much more similarity, and in general, in smaller concentrations, it can inhibit. So the relation between there is less relation between the constants. So we were not able to find direct correlation. The ability of cardiotonic steroids to inhibit and their toxicity. Next, we evaluated the impact of cardiotonic steroids on activation of kinesis, and so we found that activated at 3 hours of incubation and at 10-minute incubation at 10 nanomole per liter of bufalin respectively. Thus, we were not able to see the correlation between toxicity and concentrations activated by ERK1/2, but there is a correlation with the inhibition constants. Next, we evaluated the impact and we found that the ouabain is also activated, digoxin is not activated, and bufalin at 10 minutes of the concentration is activated, and there's no correlation to toxicity. Thus, when assessing activation of 2 kinases, it was demonstrated that it was not clear which activation could be related to neurotoxicity because we never found any direct correlation. And in order to understand the difference between the impact of toxic and non-toxic concentrations of ouabain on activation, we constructed time profiles of activation, ERK1/2, 1 and 10 micromole ouabain, and we demonstrated that activation occurs earlier, which means if a activation is observed, as you can see here, at 1 hour incubation with 10 mole incubation, there are differences at 10 minutes. The same we did with here the differences are slightly different, ERP38, and in this case activation P38 is noticeable, and in both types of ouabain. But if in general, if we look at activation profiles, activation of P38 under the influence of 10 mole ouabain, it cannot be seen, cannot be detected after 10 hours. With 1 mole ouabain, it survives. We also evaluated 1 mole and 10 mole of J and K, and here you may see that 1 micromole ouabain causes reduction of J and K activation after 6 hours of incubation, and 10 mole ouabain after 10-minute incubation causes less activation. After 3 hours, activation restores to intact condition, and then at 12-hour at incubation, J and K activation is significantly reduced. We also compared the impact of time of incubation activation in non-toxic concentrations of bufalin and toxic the digoxin. So activation of ERK1/2 occurs 1 mole bufalin after 10 minutes of incubation, and after 1 hour of incubation it decreases. At incubation with micromole digoxin, it disappears after 1 hour. The same we did for P38, and we see the same picture for bufalin. The difference is that activation, even with 8 hours activation, is preserved, though for digoxin we see activation of P38 only with 8-hour activation, incubation, respectively. As for the impact of activation of J and K under the impact of 1 mole ouabain occurs after, and digoxin did not have any impact on J and K activation. And then we studied the mechanism of activation of Kinesis depending on each other and increasing potassium concentration. We used ouabain to reduce in cellular potassium concentration with together with 1 mole ouabain. The amount of intercellular potassium not only does not lead to, but may also increase activation. Also we studied the of P38 and activation under the impact of ouabain, and at 3-hour incubation, inhibition of P38 with specific inhibitor, there was no reduction of activation. Also, we studied the impact of increasing the intercellular potassium concentration, and activation of P38 we found depends on increase of potassium concentration at various incubation times. And we also studied ERK, the impact of ERK2 activation, and as we can see, with 3 hours incubation, activation of P38 does not depend on activation of ERK1/2. And we also studied the impact of potassium on J and K activation under the influence of ouabain, and we demonstrated that intercellular potassium there's no effect of ouabain. JNK is not activated. Also, when inhibiting ERK1/2, JNK was not activated, and with inhibition of P38, it was also not activated under the influence of 1 mole ouabain. So we can conclude that at 3 hours activation does not depend on activation of P38 or increase of potassium concentration within a cell, and concentration of P38 does not depend on amount of potassium inside a cell, and after 12 hours of activation, as was shown on a diagram, but at that its inhibition does not lead to activation of P38, and activation of P38 depends on concentration of and inactivation of JNK depend on all 3 factors: activation of P38 and increase of potassium concentration inside the cell. Then we studied the impact of concentration of potassium inside a cell on ouabain toxicity, and we found that it does not protect against impact of 10 mole ouabain, thus inhibiting kinase ERK1/2 protects from impact of 10 mole ouabain and protects both with a simultaneous addition and when added 3 hours after 3 hours of incubation of 10 mole ouabain, which can mean that exactly lone activation leads to, and under the influence of 10 mole ouabain, inhibition of ERK1/2 did not protect the culture. We studied toxicity of ouabain on activation of P38, and we found that when the impact of 10 mole ouabain on the culture, inhibition of P38 does not lead to an increase of resilience of the culture. Moreover, so P38 of 2 mole leads to reduction of resilience, while addition of 1 mole ouabain reduced this effect. So what can we say in conclusion? Under the influence of non-toxic concentrations of KTS, such as 1 mole ouabain or bufalin, ERK1/2 is activated. That doesn't depend on activation of P38. There is activation of P38, which, depending on incubation time, may depend, but depends on concentration of potassium inside the cell, and JNK kinase activation depends on ERK activation, P38, and increase of potassium concentration inside the cell. And we may conclude that lone activation of ERK and more obvious activation and deactivation of JNK kinase after 6 hours of incubation. So this condition of cardiotonic steroids, which do not lead to death of neurons, and the impact of ouabain and digoxin leads to long activation, ERK activation of short activation of P38 and deactivation of JNK, and it can be divided into fast up to 1 hour and slower after 8 or 12 hours cultures, which leads to the death of culture neurons. Let me not read the conclusions. Yes, thank you. I think we were all able to study your conclusions. Well, dear colleagues, do you have any questions for the applicant? The endogenic KTS. What do you have any data about brain for non-cellular space? Is it? Yes, we have data for brain of cardiotonic steroids of ox hypothalamus, so they can circulate in the blood, and their concentration usually is 1 nanomole per liter. So they never reach toxicity levels. Here, there's one thing. At various, their concentration can reduce. So if we talk about such somatic pathology, chronic kidney failure, the concentration can increase by several orders, and in the brain as well. At the moment, it's hard to investigate it in the brain because you need lots of material, and it's hard to get samples, but blood is easy. In animals, when modeling bipolar disorders or depression, for example, when modeling amphetamine, introducing antibodies to cardiotonic steroids, it reduced their efficiency of amphetamines. So there is, at the moment, we are researching this. But at the moment, it is still the mechanism is the synthesis mechanism is not completely clear, and all their functions we have not been identified. And our group is working on that, and many other groups are working on that. And so, if their concentration increases, it can reach 10-20 nanomole per liter. But how is that related to 10 micromoles, which are toxic in ouabain, for example, in red neurons? The thing is that for human neurons and neuron cultures, toxicity concentration starting at 10 nanomoles per liter and higher, because in humans sensitivity alpha-1 isoform is much higher, is by several orders. So it's comparable to alpha-2 and alpha-3 sensitivity, unlike rats. But in rats, it's easier to divide the effects. For example, we add and we see the inhibition effect of alpha-2, alpha-3, but alpha-1 is not affected. So we affect activity of alpha-1. Yes, thank you. Do we have any questions? Not yet. I have another small question. You studied different substances. Maybe there is any connection between the structural peculiarities and toxicity on myocytes, maybe not on brain cells, but on some structural. Yes, let me go back. It was in the introduction. Here you may see the structure of cardiotonic steroids depends on the depth of KTS binding pocket, where cardiotonic steroids enter from the outside, and the amount of cardiotonic steroids, they can be a steroid and ring. The cardiotonic steroids as a 5-member ring and 6-member ring, and the specificity can be determined by lactonic ring or the amount of remaining sugars, residual sugar present in the molecule from 0 to 3, and the less residual sugar there is on the one hand, then on the other hand, through greater variety in structure, more higher specificity can be achieved. And I have a small question: if there are structural correlations, or maybe new synthetic analogs are being created, maybe higher efficiency, maybe lower toxicity, and on the from the point of impact on central nervous system, do you see any potential? Yes, at the moment there are many cardiotonic steroids, and some of them can replace digoxin, because digoxin indeed had significant side effects, which initiated the study of cardiotonic steroids, and indeed they are used already. But as far as I know, the study is in system injection. There are side effects, but not everybody is studying the action mechanism, though some groups are investigating the mechanisms on various levels. But here it's important to understand how the structure is related. Bufalin, as Applicant, has no residual sugar, but ouabain and digoxin, on the opposite, they contain residual sugars, 3 residual sugars, and how this activation depends. In this work, I have not included all signal cascades and protein kinase C and others, but depending on. So there is the task which hopefully we were able to solve in my work, how the structure of cardiotonic steroid determines how it affects neurons, and how the process it initiates lead to death of neurons and not. Thank you. Alexander Vasilievich, more questions? Okay. May I ask a question? So please, tell me please, neurotoxic activity of ouabain. It's known that its main manifestation is in induction of neuron necrosis or inflammation. Did you find any precursors of neuron inflammation, or apoptotic cascade, and what is their relation? What's the relation of apoptosis and necrosis and neurotoxic ouabain and other cardiotonic steroids? And my second question, just for me to, is it the same as bufalin? Sergey Mikhailovich, thank you for your question. Let me start with the end. This is a different bufalin is bufaginin, extracted from Bufa marinus. As for the death of neurons by necrosis and apoptosis, high concentration of ouabain is 80 micromoles per liter. They lead to mixed death. There is death of neurons at which we see symptoms of necrosis and apoptosis. And of course, it would be interesting in the future maybe to find out neurotoxic activity, which type prevails. But since I have my task was to find the minimal concentrations, I believe that should look at in this work at what apoptic mechanisms are active. So I looked at the ratio of mitochondrial proteins, initiated, and I looked at activation of pathological changes and other signal cascades, different stress conditions, cell damage. So yes, earlier we demonstrated that necrotic component is there. Are you satisfied with the answer? Yes, I am. Viktor Matveevich, do we have more questions? No. Okay. We have not received any. Yes, there is a question. What other cells, in addition to neurons, can be destroyed by ouabain? Sorry, could you please repeat your question? Alexander Vasilievich, please tell us, besides neurons, what other brain cells can be destroyed by ouabain? Thank you. Besides neurons, in brain, glialic cells, and the effect of ouabain prevents inflammation and also cardiotonic steroids can affect the endothelium cells, brain capillary, but at the moment it's hard to say how exactly this happens. Indeed, there are studies which indicate the cardiotonic steroids can reduce inflammation, I see, thank you. Do we have more questions? Council members? If there are no more questions and we don't have any external reviews, we can proceed to reviews. Maybe the applicant may have a seat. Please have a seat, thank you. Then we will proceed to reviews of the council members. Maybe Sergei Mikhailovich will start. Dear council members, dear attendees, dear colleagues, the relevance of the problem is obvious because this sodium pump is an important driver of processes in all body cells. So the cardiotonic steroids and this study is extremely relevant today. Earlier we studied the effects of ouabain and other cardiotonic steroids were not studied from the point of neurotoxicity. Besides the cardiotonic steroids, in low concentrations, nanomole concentrations may also act as hormones modulating activity of the cells, and cardiotonic activity in low concentrations is connected with the fact that in the muscles, heart muscles, additional potassium is released, and this leads to results from neurotoxicity, and this regulatory function today is being explored, and it will be of the physiology of the cells. So this determines the relevance of the work, because first of all digoxin and bufalin have not been studied and for the point of their neurotoxicity, and the study of mechanisms of intercellular regulation of cardiotonic steroids, especially considering the fact that they are present and they can be endogenic and can perform regulatory functions, but in different pathologic conditions their concentration may change, which can lead to toxicity manifestations. As a result of his study, the author came to some very interesting conclusions. Study the activity, this potassium dependent and potassium independent, J and K activation are potassium dependent, where cellular potassium acts as a messenger, and it can regulate many processes. And besides, ERK kinase is activated without potassium emission, so which means that there can be selective ways of regulation of these cascades. From the point of theoretical significance, of course, the data obtained improve our understanding of the work of important molecules and their significance. The thesis is written is based on the traditional scheme. Starts with there is literature overview, which is cardiotonic steroids. There is results, the discussion, and it should be mentioned that the author's publications are very interesting, since these were tested and were published in leading international journals, indexed in Web of Science and Scopus, and in journals from the ministry. The methods of research are certainly adequate and relevant and well applied, and conclusions of the applicant are absolutely justified. Statistical processing is appropriate for this study. That is why I should say that the work is of very high quality. All the data discussed, widely discussed, and are of interest to general research, to many researchers. In addition to that, I would like to ask one question. One question has already been asked. That's the question of endogenic levels of KTS reach such concentrations that could be achieve neurotoxicity. And the second is purely of editorial nature. One of the conclusions on the action of cardiotonic steroids at various times is after 24 hours, 12 hours, the minimal concentration for 24 hours was given, but for 12 hours the minimal concentration was not indicated, so it leaves some field for speculation. Besides, it should be mentioned, as because the work is certainly and the results are of very high quality, and while the conclusions correspond to the tasks and the experiment results are well grounded during the discussion. So, in conclusion, let me read the conclusion. The thesis by Lopachev and Vasilievich, the study of mechanism of neurotoxic activity of cardiotonic steroids, ouabain, digoxin, and bufalin on the primary red neuron culture corresponds to the main requirements set by the order dated the 19th of November 2021 on the order of awarding academic degrees at St. Petersburg University. And the degree applicant, Lopachev Alexander Vasilievich, deserves awarding the degree of Candidate of Biology, Specialty 1-5 for Biochemistry. So this is my conclusion. Thank you for your attention. Sergey Mikhailovich, thank you. If there are no, maybe we can listen to the. Do you mind if we let the applicant answer Sergey Mikhailovich's remarks? Sergey Mikhailovich, thank you for the question. I would like to say that the effect of the exposure to ouabain, after exposure to ouabain for 12 hours, resilience reduced. Let me go back to, here is the slide I need. At 12 hours of inhibition at 10 mole ouabain led to lower culture resilience Unfortunately, this is a summary diagram for 3 mole ouabain. But for 10 micromole at 12 hours of incubation, resilience reduced, which could also indicate the development of the apoptic process in neurons. As for the question if locally the concentration of cardiotonic steroids can reduce to neurotoxic effect, I may say that in rat and mice body maybe not, but in human body, in principle, we can imagine a situation in case of certain diseases where the concentration of cardiotonic steroids and with such concentration, unfortunately, I don't have the reference, but the concentration of over 10 micromoles per liter, neurons that die or neuroblastoma cells. Thank you, Sergey Mikhailovich. Are you happy? Yes, I am. I am satisfied. Thank you. It's good. Okay, then maybe we will proceed to the next. Since all the reviews were published at the university website for preliminary examination, it's okay to focus on the key points. If so please we can make it shorter. Natalia Nikolaevna, the floor is yours. Please present your review. Dear council members, dear colleagues, we have just heard high quality presentation of the applicant, which made a very good impression. The presentation and the subject of his dedicated to the study of mechanisms is today what is highly relevant task of contemporary neurobiology. I will not give you any common facts, but what I would like to say is remind you that active study of neurodegeneration mechanisms led to new details which today are less studied, as unlike the main components of this aggregation, mitochondrial, you know all that, but many years of research of these didn't lead to efficient therapy. So anything to do with neurodegenerative processes is of high interest and is seen as potential for new solutions, new strategies of neurodegenerative diseases. Alexander Vasilievich's research is dedicated to the study of neuropathological with the cardiotonic steroids. Their role in we started to find mostly identified in the brain, and their ratio, and especially the ratio of modified forms, they change in case of various pathologies. As Alexander Vasilievich has already said, he also identified the changes in ratio of these forms, and since indeed it can be obtained via biopsy, by biopsy, human biological material can be obtained in case of strokes. Of course, it's a small work, it's just started. Not all the studies have been published, but there are also some hints that the concentrations change, especially in case of secondary damage, in case of brain injury. So it's very logical. So the hypothesis who decided to study as neurodegenerative processes, and the author set himself the task to study at low concentrations where inhibition is only partial. And for the first time, the author conducted comparative research of 3 compounds: ouabain, digoxin, and bufalin. I will not read. The author is on ERK1P and JNK in the activation and the connection with neurotoxicity. Besides, the author has analyzed the degree of inhibition depending on concentration, and this approach enabled the author to obtain new data about the impact of such an object as primary culture. Contains 5 chapters, and their material corresponds to defined conclusions, and it made it much easier, the logical transitions between the chapters. In the first chapter, in the different concentrations at various incubation times, as well as the impact in mitochondrial apoptosis of BCL2. It would make sense how apoptosis and necrosis, neurons die under the influence of minimal concentrations. For example, but of course this remark does not affect the quality. In the second chapter, the author describes the large hemispheres of red brain. The inhibition can allow to the constructed inhibition curves make it possible to estimate the inhibition constraints. Although within the scope of this work, the applicant carried out the comparison of the ratio of isoforms on red neural culture and microsomal preparations, future studies should duplicate the measurement of activity on a live culture, as a cell is a more complex system when the membrane preparation and the enzyme activity may be different from that and the membrane preparation. And in the third chapter, in chapter 3, the author explores and on activation of Kinesis and J and K in the primary neuron culture. These chapters reveal the differences between the effects of different CTS on the activation of the 3 kinases. In this chapter, the applicant demonstrates that it's not only important to demonstrate the activation of a particular signaling cascade, but also to study the process over time as a function of stimulus strength. This approach significantly improves the quality of the work performed. In the 4th chapter, results, the applicant describes the long-standing results during the exposure of ouabain 3 different incubation times. The data obtained revealed the role of potassium to the mechanisms of kinase activation, as well as mutual dependence. And finally, results and discussion describes which of the intracellular signaling mechanism whose activities affected by ouabain are related to CTS toxicity to primary culture neurons. And one of the key points is that the long-term activation of ERK12 when exposed to ouabain is associated with this neurotoxicity. However, the work does not study how the less toxic. Is this because that does not activate when exposed to lower concentrations and does not to RK2? Perhaps there are other mechanisms, but this is not an observation, but rather a suggestion for future research, because it does not affect the quality, and their conclusions are well, and it's a good work, interesting to read. And in conclusion, let me read what we have to say. The thesis by Lopachev Alexander Vasilievich on investigation of mechanisms and neurotoxic effects of cardiotonic steroids, ouabain, digoxin, and bufalin on the primary culture red neurons corresponds to the basic requirements established by the order dated the 19th of November 2021 on the procedure of awarding academic degrees at St. Petersburg University. And the applicant, Lopachev Alexander, deserves to be awarded an academic degree of candidate of biology, specialty 154, biochemistry. Articles 9 and 11 of the above-mentioned order has not been violated. Thank you, Natalia Nikolaevna. Alexander Vasilievich, maybe you'd like to respond to suggestions and remarks? Natalia Nikolaevna, thank you for your review. Yes, indeed, a cell and a microsomal preparation is not the same, and microsomal preparation, the quick signaling cascades which could be active in a cell, but on the other hand, since there was no opportunity to work with the help of which we could measure activity of potassium sodium ATPase, so we had to use this method. On the other hand, we are now working to measure the non-radiative rubidium, and so for the amount of rubidium we can decide on the and so in the future works we will be able to present the data on activity with this obtained with this method as well. And as for the apoptosis, it has already been said that on the one hand we demonstrated that at higher concentrations both processes take place, but I focused on the CBL2 proteasms. Why digoxin is is most likely here. This is about activation profiles, and in this case there is some antagonism between P38 and we see minimal activation of P38. Activation is present, but maybe there are other signal cascades also in action. Maybe it's about other activation cascades, and at the moment we are working on to study them, and protein kinase C and other. Thank you, Natalia Nikolaevna. Yes, I'm fully satisfied with the answers. Thank you. Next review, Alla Borisovna. Let's hear your review. Dear council members, I think so. I will allow myself to very briefly present my as for the relevance of the work is beyond any doubt, and the mechanism that ensures toxicity, the mechanism, and the need to optimal concentrations, cardiotonic steroids, and pathophysiological processes inside the brain, and the need to test the apoptic action, which results in effect. The effects indicate that they can affect and certainly has theoretical and practical significance. I think it's necessary to mention that Alexander Vasilievich studied tissues, primary structures, and all this. So because of that, he produced Thesis. For the first time, the author demonstrated not only but the changes in activity of. Also, the author for the first time registered of apoptic effect is the impact on and the main results set forth in profile journals included in international databases Web of Science, presented at conferences, at national international conferences, which add credibility to experts in this field. As was mentioned, the thesis is very logical, well structured. The chapters demonstrate good command interpretation, indicate that Alexander Vasilievich is a mature researcher with good analytical capabilities and own view, yet there are some. So, for example, I think the unsuccessful of the fourth provision submitted for defense were identified because it's not typical for this part of the. And besides, in the result in the results section, would make sense to present the original author's scheme illustrating the revealed neurotoxicity, yet such stylistic errors and absence of the scheme do not affect the purely positive impression. And I don't have any specific questions to the applicant, and I believe that the thesis by Alexander Lopachev, the study of neurotoxicity mechanism of cardiotonic steroids, is a complete piece of paper which addresses a relevant scientific task and the thesis corresponds to the main requirements set by the order dated the 11th of November on awarding academic degrees at St. Petersburg University, and I believe the applicant, Lopachev Alexander Vasilievich, deserves awarding the degree of Doctor of Biology, specialty 154 Biochemistry. Articles 9 and 11 has not been violated. Thank you for your attention. Thank you, Alla Borisovna. Alla Borisovna, I thank you for your review and attention, and let me tell you that so I will work on, and still working on wedding. Thank you, Alla Borisovna. So there are no further okay, I see. Very well, let us proceed to the next review. Viktor Matveyevich, welcome. Only the key. The key. Please speak into the microphone. Can you hear us? No? Is a key ferment of the central nervous system. The main function of this is maintenance of the electrochemical gradients of sodium and potassium, and disruptions in the work of this ferment are neurodegenerative, and in the 21st century is supposed to be the age of neurology. In the central nervous system, there is a number of functions not immediately connected with the pump functions, which include regulation of the work of other membrane proteins, activity of cellular signal cascades. At the moment, there is a number of facts that this ferment also receives cardiotonic steroids, or with the help of various experimental models. It was demonstrated that steroids may affect efficiency of sodium and potassium, inhibiting the work of is complete in suspension of granular cells. With 1 mole ouabain leads to accumulation of glutamate and hyperactive inotropic glutamate receptors, which leads to high concentrations of sodium and potassium inside neurons, and which lead to the death. But at the moment, there is no full picture. We don't have a full picture. Besides available information about neurotoxicity of steroids, mostly describes the effects of ouabain, as well as other steroids, the study of neurotoxicity of which is also relevant. From that point, the thesis by Alexander Vasilievich Lopachev is dedicated to the study of mechanism neurotoxic activity of cardiotonic steroids, ouabain, digoxin, and bufalin on primary structure of red neurons is certainly relevant. I will not describe the structure of the thesis. It has already been described, yet the literature overview fully provides detailed characteristic of ion pump and steroids as specific inhibitors of this, and outside the central nervous function in physiological central nervous system. What is important? The work is built according to the and in the final chapter, the author demonstrates neurotoxicity of high concentrations of ouabain is connected with prolonged activation of ERK12, resulted from activation of P38 and doesn't depend on high concentration of sodium 2. Their conclusions are well grounded and fully correspond to the text. The results evoke no doubt and statistical correct with the use of contemporary methods. The author published 5 papers, including 4 papers in peer-reviewed databases Scopus and 4 in Web of Science. And in conclusion, the experimental study conducted on primary culture of rat neurons fully disclosed the new mechanism on neurotoxic activity of cardiotonic steroids, ouabain, digoxin, and bufalin. In general, the work has high relevance and has fundamental significance for the understanding of steroid mechanisms in physiological processes in the central nervous system. The thesis by Lopachev and Vasilievich on the study of mechanisms of neurotoxic activity of cardiotoxic steroids, ouabain, digoxin, and bufalin on primary rat neuron culture corresponds to the main requirements set by the order of the 19th of November 2021 number 11181/1 on awarding academic degrees at St. Petersburg University. And the applicant, Lopachev Alexander Vasilievich, deserves awarding the degree of candidate of biology, specialty 154 biology. Articles 9 and 11 of the above-mentioned order has not been violated by the author. Thank you. I don't have any critical remarks, the questions have already been asked today. Thank you, Viktor Matveyevich. Then let me read my review. I'll try to be brief. Though they are still in demand since they are administered with chronic heart failure and other different conditions. And today, when the new virus arrived, especially in post-COVID patients, the study of cardiotonic steroids became even more relevant, especially of their side effects of heart glucosides. The molecular foundation of steroids have been studied for many years, but these works mostly focus on analysis of steroid effect, and the effect on central nervous system have not been studied in detail and requires additional study, since the issue of side effects in different patients with nervous system is not been solved. It's also important of endogenic steroids and the study of activity of biosynthesis of these compounds in the brain, and the dependence of this process on is also has not been explored. So the theme of this study is certainly relevant in question. The validity is of the conclusions is quite high, since the methods correspond. The work has been tested at various levels, and results were published in journals recommended by the Ministry of Education and Science of Russia, and 5 articles published, 4 in articles indexed in the scientometric databases Web of Science and Scopus, when one which is one of the first quartile article, which is of high value today. In general, as I will not go about explain novelty for the first time, comparative analysis and neurotoxicity of several steroids, ouabain, digoxin, bufalin, obtain new data demonstrating the connection between these steroids and activation of kinesis and their neurotoxicity. The author obtained new data which explain the effect of ouabain and activation of signal cascades in general. The theoretical significance is in the fact that the data neurotoxicity improve understanding of the role of endogenic steroids, specific features of regulation, the reasons for dysfunctions in neurodegeneration. From the practical point, the results can be a contribution in the study of minimization of on neurosystem. And here I think is minimal neurotoxic concentrations. The data is the constant inhibition constants, and that on Kinesis. The thesis contains 253 pages. Introduction, literature overview, materials and methods, results, and the thesis has 48 diagrams, 340 sources. I will not go into details. Explains relevance of the topic, explains novelty, theoretical and practical significance. A literature overview contains mechanisms of cardiotonic steroids, specific inhibitors of ATPase, discusses articles dedicated on mammals, analyzed understanding of ATPase and physiological and pathological process in the central nervous system. Methods and materials have already been discussed here. The chapter contains 11 sections which explains different biochemical methods. The results contains of 5 compares them with existing literature. So I will not go into details here, because this has already been discussed in detail, and some minor comments maybe. The values were missing from some diagrams, but that is not. There are some typos, but I think some wording, for example, in scientific novelty, I will not go into detail. So all this you can read in some suggestions, but the conclusions are well and correspond to the set tasks. And in conclusion, the summary is also quite good. Thus, we may conclude that the thesis by Lopachev Alexander is a high quality work which addresses neurotoxicity of cardiotonic steroids. In general, is on the role of cardiotonic steroids and can be used in medical biological studies to reduce toxicity of the heart system. So I have one question. So what do you see the prospects? So what are you going to do next? And the conclusion, the thesis by Alexander Lopachev, investigation of mechanisms of neurotoxic of cardiotonic steroids, ouabain, digoxin, and bufalin on primary culture rat neurons, complies with the basic regulations established by the order number dated the 11th of November, number 111181, on awarding academic degrees at St. Petersburg University, And the applicant, Lopachev and Vasilievich, deserves to be awarded the degree of candidate of biology, specialty 154 biochemistry. Articles 9 and 11 of the above-mentioned order has not been violated. Thank you. Olga Valerivna, let me thank you. I will certainly consider all your comments, and as for the repetitions and as for my future activity, right now I am working on I'm studying neurotoxicity as well as an interaction of sodium-potassium ATPase and cardiotonic steroids, and so that it includes their connection with neurological diseases such as Parkinson's disease and neuropsychiatric diseases, and the possibility of transition from one disease to another, and also the role of potassium ATPase in the neuron development, what has been done besides that, and the dopamine and our models we will in rats, and transcriptome research, gene expressions which ouabain may cause, for example, changes of gene expression, growth of neurites, and may affect maturation of neurons, and the metabolic processes, neurons, and in the future we are going to study this field and continue this study of role of cardiotonic steroids in pathologies of the central nervous system, dopamine energy system, and ideally I would love somehow to approach the question of their synthesis and regulation of synthesis in the brain, but this is a dream. I hope your dreams will come true. Please have a seat. Okay, then, dear council members and dear colleagues who are not members of the council, do you have any questions, perhaps? No? Is anybody willing to speak, express his opinion? Not yet. Very well. Have we received any external questions? No? Okay, in that case, dear council members, is everybody satisfied with Alexander Vasilievich's answers? Yes? Very well. Then, is any of the council members willing to speak? Does anybody have questions regarding the procedure related to the remote access mode, or maybe any other? Sergei Mikhailovich, you have any questions or comments regarding the procedure? No, I think everything went very well, and thank you. Natalia Nikolaevna, you have any comments regarding the procedure? No, I don't. Well, no interruptions, so all was very well. Alla Borisovna, what is your opinion? Do you have any remarks regarding the procedure? Remote access mode. The microphone, please. Turn on the microphone. First, we cannot hear you. No comments. That's good. Thank you, Viktor Matveyevich. Probably we have. You have any critical remarks, Viktor Matveyevich? No. It works very well. Thank you. Okay, then, if nobody's willing to speak, then maybe the discussion is over and we can proceed to the next item. So please have a seat. Let's proceed to the next item, which is voting. Dear council members, we have the opportunity to take a break, or we don't need a break to discuss the results. Do we need a break? I think we don't. Let's finish. Then let us vote, proceed to voting. Oh yes, Alla Borisovna, perhaps you could you say something? Yes, now we can hear you. Thank you. Thank you. Then it's 4:45. My watch says 4:45, and let us proceed to voting. Each council member has to express his or her opinion individually. We can all see and hear each other. Let's proceed to individual voting. Here we simply have to answer briefly. We just give our opinion, right? I'll say then, Sergei Mikhailovich, what is your opinion? I think that this is very high quality work, world level thesis, and so that deserves high assessment. So I will vote for. Natalia Nikolaevna. That's an excellent thesis. I have no comments. The author fully deserves awarding. I will vote for. Alla Borisovna, what is your opinion? I support this work. The work is interesting, very relevant, and as I support the work, I vote for awarding the degree. Thank you. Viktor Matveyevich, I support the positive assessment. This is serious fundamental work and highly relevant, so I support. I also liked this thesis and the applicant's speech and answers to questions, so I will vote for. Now we have to article. Now we have to say that, do I have to read Article 11? Thus, dear colleagues and guests, let me inform you that out of 5 council members, 5 voted for, no one voted against and no one abstained. So 5 council members are for awarding the degree. So the decision to award to Lopachev Alexander Vasilievich the degree of candidate of biology, specialty 1-5 for biochemistry, has been made. Your closing remarks, Alexander Vasilievich. I would like, first of all, to thank my academic supervisor, Tatiana Nikolaevna Fyodorova, for her guidance and assistance at all the stages of for preparation of this work. Also, I would like to thank other students and doctoral students of the laboratory of experimental laboratory where this work was written, and for their time and attention. I'd like to thank the council members, Olga Valerina Shamova, Viktor Matveyevich Klimenko, Natalia Nikolaevna Ninkina, Alla Borisovna Salmina, and Sergey Mikhailovich Antonov, as well as all my colleagues from St. Petersburg University for their help and support. Thank you. Thank you, Alexander Vasilievich. Also, I'd like to say that unfortunately the academic supervisor was absent for a good reason, but we have her positive review. The review of the on the applicant's work is fully positive. There are no comments, no remarks. Everything is very well. So now I can congratulate the applicant, Alexander Vasilievich Lopachev, with being awarded the degree, and wish him further success. May all your dreams and all your wishes come true. We sincerely congratulate you on this remarkable occasion. It was a brilliant defense and the positive decision. At that, I declare the session closed. Thank you, everybody, for your time, and those working in remote access mode, and to all the attendees. Thank you, everybody. Please stop the live broadcast. Thank you very much, everybody, once again.